right, so just like gauge what you guys are interested in. How many of you guys have like backgrounds in biology or anything like of that nature? Okay, so not really. Okay. So in that case, I'll focus more on like the machine learning technical aspects than like the biological application. So this paper here is recent work that we worked on in Victor's lab. It's a prediction model that takes in a protein-ligand crystal structure and predicts the binding affinity between the protein and the ligand. And it uses two types of architectures that are fused together. Let me see if this is clear. Yeah, so you guys learned about convolutional neural networks, right? So fundamentally, they process some data that's represented as an image. Now, you could represent a lot of things as an image, including a protein or a protein-ligand complex, by basically looking at a protein-ligand complex in 3D space and building a grid around and within that protein, such that you can assign, let's say the grid is made up of small cubes. So it's a big cube made up of smaller cubes. And you assign each atom to whatever cube contains its center, which is what you see here. Let me actually probably draw over here. Actually, I'll try to stay within the frame of the thing. So I'll go over there. All right, so like imagine you have like a protein like this or something, and then you build a cube that's made up of like smaller cubes like this. Not an amazing drawing, but say like you have an atom here and it's in this cube. So you have that cube represent that atom. And what this leaves you with is a 3D structure. So you could think of this as a matrix that's 3 dimensions, where each element in the matrix corresponds to that atom. Okay, so now you could represent each atom by a vector of chemical properties. So rather than this data type being 3 dimensions, it turns into a 4-dimensional object. So you could have each vector having elements of things like atom type, partial charge, molecular weight, things like this. Does this make sense? Okay. And you guys could like interject at any moment, ask me a question. So once you have this data set up in this way, you perform a traditional convolution. I'm sure you guys probably learned it for 2D images. That's usually how it's traditionally taught, but it's the exact same process. But rather than the filter covering 2 dimensions, it's covering, in this case, 4 dimensions, but it's the exact same process. So yeah, you can think of this as just like a pretty traditional convolution with some additional technical features that assign importance to each one of the features themselves. And the output is just a 1-dimensional float, which is the binding affinity. The training data is, the input data is protein-ligand PDB files, and they're each labeled with the PKD, binding affinity, experimentally determined. So yeah, you fit this model to that data, and the results turn out to be pretty good, which is kind of interesting because it's not super intuitive to think of proteins as images, but it actually works. And CNNs are applied to a lot of different things in biology and also obviously image processing and things like this. And they have their niche advantages. Yeah, but almost all of it is related to image processing. So this is one of the 2 architectures that's in this model. The second architecture, which is fused to the CNN, is a graph-based model. So the input data is exactly the same, but rather than representing it as a 4D grid, it's a traditional graph. So you have nodes and edges. So the nodes are atoms, and the edges are bonds between the atoms. So it's very intuitive. And each node is represented as a vector, similarly to the CNN, where each element in the vector is some chemical feature. Now, this is a straightforward model. You perform message passing between the neighboring nodes. and you're left with a prediction of the binding affinity. Now, we fuse these two models. Does anyone have any questions about this, by the way? Here? Good. Okay, cool. So we fuse these two models and we benchmark it on the most popular benchmark for predicting protein-ligand binding affinity. And it's currently the state-of-the-art model in the field. It's this one, HAC-Net. This is like just showing RMC. It's like
I have a question about the voxel.
Mhm.
Representation.
No.
Would you do, is like the resolution of each atom in each voxel necessarily, I guess, accurate? Because like like, what do you do if an atom doesn't fall exactly within a given voxel?
It's a great question. So each voxel is 1 angstrom cubed. We assign the atom to the voxel that contains center. Now the center is just a point, it has no bottom. So like it has to lie in exactly one voxel. Right? Because it's just a point. So it's not like, it definitely could be the case that if you're looking at the van der Waal radius, it's going to overlap. But we just take wherever it's center, we assign it.
Okay.
And it's a good question because this does lose some information in a sense, because it is like,
You're forcing them to go into.
Yeah,
the
like-
box.
like say like the atom center is like here, right? We sort of assume it's like there, you could say, in theory. But yeah, so it's a good question, but yeah, that's how we implement it. Some groups that use CNNs for things like this, what they'll do to sort of try to alleviate this kind of thing is they'll introduce noise, like Gaussian noise, to the whole grid to blur it so as to not have the model assume that that's a precise assignment. Do you know what I mean? So they'll sort of like blur the whole thing.
Alternatively, couldn't you just use much finer voxels? I guess it gets more expensive.
Yeah, so it's already 500 million parameters roughly. You could use finer voxels, but that would, assuming you had infinite compute, that wouldn't necessarily be better because then you would likely overfit the training data.
Okay.
Because there's a balance, generally speaking, in machine learning, there's a balance that you need to find between number of data points and the complexity of those data points with number of parameters in your model. If the ratio is off, if you have too many parameters to data points, you're going to overfit the training data. If you have too many data points to parameters, you're going to underfit. So if you were to decrease the voxel size, make it super precise, it's likely going to make it very difficult for the model to then predict binding affinity for structures unlike those in the training. Does that make sense? So it is a balance. Yeah, it is a balance. And we tried using different voxel sizes and found 1 was pretty good. Maybe like 1.2 was better. We didn't try. We tried like 1, 2, 3, 4.
Is this just the heavy atoms?
Yeah, it's just the heavy atoms, yeah, no hydrogens. Cool. Okay. Yeah, so we apply this to the benchmark that everybody uses. And we get the best results. So it's this one, HAC-Net here is our work. Yeah. Overall, it's the best model.
And what training set they use?
We use PDB bind, the 2020 version. This is like the most popular public data set for protein ligand structures, if you guys are familiar. If not, yeah, you can check it out. But the thing with public data sets that's pretty cumbersome is that, you have different labs contributing data and it all gets combined into one data set. And the experimental methods themselves have variance. So you can't assume, so basically it's been shown like you could have the same exact complex, but the biochemical assays were done at different labs, the values won't be the same. So there is this intrinsic noise in the data, which makes it difficult for these models to become perfect. But yeah, that's a digression. So
in that table, what was the difference between the two HAC-Nets on there, right? There's HAC-Net, double dagger, and then HAC-Net.
so the training set is slightly different.
Okay.
So this is like all the stuff. Okay, so this is complexes from the 2020 and 2016 data sets they have. This one is just the 2016. And we show like, yeah, it's still pretty good even with way less data. That's the point of this. So, yeah, I'm not gonna, okay, you know, this is actually probably useful to mention. So one pretty major problem within this area of study is that what people will do is they'll make sure their model can do very, very well on whatever data set they're benchmarking, but then it's like no good at other stuff. So like they'll basically they'll basically like use the benchmark as the validation set and like choose the parameters that do best on this. Super cheap. And there are other there are other intrinsic problems with this benchmark, and there are a few papers criticizing it. For instance, if you train on just the ligand, no protein, you can still get like a 0.2 correlation on the prediction, which is crazy. Like you should get exactly 0. So to demonstrate that our model is able to generalize, like truly generalize to new data points, we created additional training test splits that attempt to alleviate some of these biases. So one thing we do is we create a training, validation, and test set split where the data points differ significantly with respect to protein structure. So the protein structures in the training set are very different than those in the validation set, are very different than those in the testing set. We do an analogous thing, but for protein sequence as well. And then these are the results for that. It's actually still pretty good. So when the data points in 3 sets with respect to protein structure are very different, we still get roughly 0.8 correlation, which is pretty good. We didn't cite other papers in this because it's like, it would almost be like a hit piece, to be honest. It's like something, but they're getting like 0.4 more or less. And Rafi and I, the other author of this, we attribute the ability to generalize to the fact that we have 2 different architectures in the same model, claiming that whatever errors are due to the graph network are somewhat combated by different errors in the convolutional neural network, and it's sort of like merges them to converge more toward the experimental value. So the sequence-based splits we also do well. And then we also do something similar, but for the ligand fingerprints. And yeah, we did 10-fold cross-validation with the ligand fingerprints. And we did decently well, like 0.7-ish correlation. And this is just a bunch of generalizations. So
can you say something about how you tune the hyperparameters for your, for each channel? Is that
so
how you went about, optimizing the hyperparameters? What was your strategy approach?
sure. So for the CNN here,
yeah,
where is it? So for the CNN, our loss function was just traditional mean squared error. So you have one float, which is the true value. You have one float, which is the prediction. You calculate the mean squared error. For each batch, you have a list of these, and then we used the RMS prop optimizer. Which have you guys heard about the Atom optimizer? It's pretty common. So yeah, it's basically performing gradient descent with some other fancy stuff going on where they're calculating things like the momentum and things like this. But Atom is the most popular, I'd say, optimizer. We tried a bunch of different ones. RMS prop worked the best. It's very similar to Atom, but it doesn't have the momentum component. So we use RMS prop for this. But for the graph network, we use the Atom optimizer. And that shows to perform the best. And I actually want to make one additional point. I think this figure is in the main text. It may be in that something. Okay, I think it's in that side. Let me see if I can grab this quick. It's in the main text. Oh, no, it is in the main text. So. Ooh, that's. Okay, that's decent. So, okay, so. The major like innovative claim we have, aside from the fact that we're merging two architectures, is that within each architecture, we incorporate different forms of attention. And what attention is in a machine learning context is having a model learn weights that are assigned to specific components to basically tell it to focus more in like lame, non-technical terms. So in the CNN, we have for each atom, we have a vector, right? In the CNN, we learn attentional weights to apply to each element in the vector, such that each feature is weighted based on its importance to the prediction of finding things. In the graph network, we do basically the same thing, but we also weight the importance of the neighbors with respect to the central node. And now on the left here is our normal HAC-Net model, and on the right, vanilla HAC-Net is the same thing with the attention mechanisms taken in it. And what we see is that it's decent, but definitely worse. It's actually significantly worse than the HAC-Net with the attention mechanisms.
I have a question. I might have seen a paper that does protein-ligand binding affinities that combines CNN and graph network.
You know what it's called?
No, but it might have been out before this. I don't know if you saw one.
I know what you're talking about.
Did that have attention?
No.
Yeah.
You're talking about this. Is it this?
Yeah, yeah. So the
You
difference.
want to see something? You guys want to see something fun?
Yeah.
So they say model parameter files are available here,
Oh, we know that.
right? That's not uncommon.
No.
So. Okay. Unlike that work, all of our code is open sourced. And here's just a quick demo video. So Rafi and I put together this notebook to make this very easy for people to use that don't know how to code. So you just run this notebook. The video, for some reason the video is pausing. I don't know why this video is pausing, because it always works. This is just like a demo video here. Okay, here we go. Yeah, so you just load in, you load it, you type the path to your protein file, ligand file, you run it, and then in a few seconds, you get the binding affinity prediction and a picture.
Amazing.
So, and yeah, you guys could run this if you want. You just go to the note, you go to this GitHub. It's going to tell you how to do everything. The notebook is here. So yeah, it brings you a notebook, you put your files. Done. Okay, so we were hoping that, like open sourcing this and making it easy for people to use, people to actually use it. So like a month ago, this group used it and they found a potential drug for this G protein coupled receptor that they say may, like people like to hype up. They say it may be a potential cure for MS and some types of cancers. So it does inhibit this protein, which is implicated in those things. So that was pretty cool to see. And then this group identified a potential drug for antivirulent staph infection. So people are using this rapid to see that. So does anyone have any questions? Because I'm going to be done with that.
And the number of filters and how many convolutional layers included, how did you tweak all that?
Oh yeah, so in CNN, Okay, so we've got like 1, 2, 3, 4, 5, like 6, 5-ish layers. We tried 2, we tried 3, 4, 5, 6, 7. And this is like a general statement. The more layers you have, the more parameters, the more likely it is to overfit. The less you have, the less likely it is to fit the data. So through trial and error, to be frank, we just tried different sizes and we found that this resulted in the best performance on our validations. Yeah, and the same can be said for the graph network.
And then the skipping additions,
The ResNet.
with ResNet.
Yeah.
You tried also skipping 1, skipping 2, or skipping more, and you concluded that this is the best performing.
Yeah, this was the best. So are you guys familiar with the ResNets?
Yeah.
Yeah, so, yeah, they nodded their head. So basically, what's really cool about ResNets is, so basically, if you throw in a residual connection between 2 layers, what you do is you take the output of the layer before and sum it to the input to that layer. And what this does is it allows models to basically learn with way less parameters than you would otherwise need. Because it's sort of like, if you're thinking about the backpropagation process, rather than having like a single timeline, this is one way to think of it, rather than having like a single timeline going backwards, it sort of creates like another timeline. This is one way to think of it. Yeah, so we didn't invent these, but ResNet is, I think, like 2017 or something like that.
Combined.
And they perform the best at the time on image classification. So yeah, they're very popular CNN architectures. Yeah.
Beautiful.
Cool. So I'm gonna now move on. I guess I'll start going a little faster. So okay, anyone have any more questions about it? Okay. So after that, I've been working on a generative AI-based framework where the objective is to take in a drug candidate that's cardiotoxic. But aside from being cardiotoxic, it's great, but it's cardiotoxic. And re-engineer it to preserve the pharmacological activity while reducing the cardiotoxicity. And by cardiotoxicity, specifically what I mean is inhibition of a specific cardiac ion channel that's important for regulating your heartbeat. That channel is called the HERG channel. So the way this works, don't be like scared. It's pretty straightforward. So there are 3 prediction models here. 1, 2, 3. Each of these takes in a small molecule as a smile string, and it predicts the activity against one of these 3 cardiac ion channels. Okay, so now that you know that, this initial model is here on the left is a transformer. It's the architecture of ChatGPT. It's like the architecture of almost all the natural language processing models. It's like the big deal of generative AI right at the moment. And what we use it to do is generate smile strings. So yeah, I'll get into the technical details if anyone's interested. But basically, you provide it with a molecular scaffold as well as a set of physical chemical properties, and it generates molecules with that scaffold and with those physical chemical properties. Things like log P, like solubility, molecular weight properties like this. And then you then filter that initial molecular library with these predictive models based on the desired activity against each of these channels. And this leaves you with a pool of molecules. So the way this works, practically speaking, is say you have a drug that's cardiotoxic. You initially generate a molecular library conditioned on physical chemical properties of that drug, as well as the scaffold of that drug. to generate a library of compounds that have the same physicochemical properties or very similar and the same molecular scaffold. And then you filter that library based on desired activity against the cardiac ion channels. Then we create a descriptor vector for the initial compound and each of the refined candidates and compute the cosine similarity between the descriptor vectors to identify the refined candidates that are most similar to that input molecule. And yeah, so we're working on this with Pfizer at the moment, as well as potentially BI, right, at some point. Yeah, so I think this may be interesting. So the way the data is represented for the predictive models is in 3 forms. One form is a graph, straightforward. Another form is a fingerprint, which I'm sure you guys are familiar with. And then the third is this pretty cool representation where I train a transformer on the task of, here, I'm gonna need to draw it. Okay. So say you have a smile string, just like something super simple, I don't know. All right. Here's your smile string. We train a transformer that takes this, it randomly masks tokens, and then the task is to predict what is supposed to be there. That's the task. Okay, so it turns out if you do this on millions of smile strings, you wind up creating a model which is forced to develop an incredibly rich interpretation of molecular structure. Because in order to, take in a huge smile string and know what goes where, you're forced to learn a little bit about what's going on. So we train this model, and then what we do is we take the trained model, and for a given smile string, we pass it through, and we extract a vector right before the output layer. Won't get into the details unless you're interested. And we use that vector as a feature representation of that smiles to then train the discriminative, if that makes sense. So we have these 3 representations, and in a pretty traditional way, we use these to predict activity against a given cardiac ion channel. So you guys aren't that interested in bio, except for you, so I'll say like one thing. We apply it. There's this FDA-approved drug called Pimside, which causes fatal arrhythmias, all sorts of problems. Nonetheless, it's FDA approved. The binding affinity, or rather the PIC50 to the hERG channel is 8.5, which is more than 3 orders of magnitude more than what would be considered a problematic hERG blocker. So we think, okay, let's try to make this a little bit better. So we apply the method to this, and by the way, it takes roughly 1 minute to apply the whole framework. And in that 1 minute, we generate 100 refined candidates. Among those 100 is this other drug called fluspirulline. And the reason it's interesting that this is here is because it's of the same class of drugs as Pimside, meaning the pharmacological activity is highly similar, but the activity to the hERG channel is 3 orders of magnitude less. So it worked. Yeah, code's available here, tells you how to run it. Anyone who's interested, you type in CardioGen AI, you'll find this pretty easily. Yeah.
And then the generation is by sample and then post-selection, or how do you, or you condition?
Both. So the generation is conditioned on the scaffold and a vector of physicochemical properties of that input molecule. And that gets you an initial molecular library. And then you filter that library based on, with these predictive models, based on the activity you want against each of the cardiac ion channels.
Beautiful.
Yeah. So I've got one more thing I could talk about, if you want me to, okay, does anyone have any questions about this? Yeah,
So what did the other 99 candidate molecules look like? Cause I assume the model thought they were all about the same level of efficacy.
wait, you assume the model what, sorry?
Well, I was thinking that like the model would output all 100 and think that they were all equally good.
It does, yeah,
Okay.
yeah.
So how did
Oh,
you choose that particular?
I chose it,
Yeah,
yeah, okay, yeah, so. The 100, the model are all fine.
Yeah.
They're ranked by similarity to the initial one. And this one is not the most similar, so I don't want to create the potential viewpoint that it is the most similar. But I looked, me personally, like now no longer the method, I looked at these 100. And I basically what I did was a database search, like are any of these known drugs? And this one came up.
it's already in existence, huh?
Yeah, it's already in existence. Yeah. But what I found was, and there's a figure for this in SI, is that this region here is what's problematic. So this is what's being edited. And the model figures out that this is the problematic region. Yeah. So, yeah. Any other questions? Okay, cool. Okay, so this is the last project I'll talk about. This is not in itself a model, but is actually a method to be applied to a generative model. It's an active learning method. So the point of active learning is if there are so many potential data points that it would be unfeasible to label and train on all of them because it would take too long, you could come up with a clever way of choosing the ones you want to train on and still acquiring good performance. That's the point of active learning. So before we did this is recent also. Yeah, this is published January this year. So there are a good amount of models in the literature that utilize the transformer architecture to generate molecules. Similarly to what I just showed you, like that's not novel. We weren't the first ones to have that generative component. And a lot of these are able to generate molecules with specific drug-like properties and things like this. But practically speaking, when you're generating drugs, arguably the most important thing is that it binds your protein target. This is like the thing you want, the main thing. There are other things that are important as well, but you need it to bind your protein target. And there, at the time, and I believe it's still true, there are not good methods for taking one of these generative models and aligning it such that it generates drugs that will bind to a protein you're interested in. So what you could do in theory is, generate 1 million molecules, dock each of them to a protein target you're interested in, and then, score the docking poses, and then further train on the ones that obtain good scores. But this would take forever. Like it's not feasible to dock 1 million compounds or anything close to that. I mean, if you got like thousands of GPUs, sure, but like this is like very difficult. So we came up with a method that allows you to fine-tune one of these generative models toward a protein target while only having to retrain on 1% of the generated molecule. So see if I can get a high resolution version of this. Nice. Okay. So the first step is to pre-train a transformer decoder like the one I described before. And what this allows you to do is you just generate a ton of smile strings. Simple. Okay. Then you generate a ton of smile strings, 100,000 we generate. Then for each smile string, we calculate a descriptor vector using RDKit, which is just a cheminformatics software package. It's super easy, it's very fast. And yeah, so we convert each smile string to a descriptor vector, where each descriptor is a physical chemical property, like molecular weight, number of hydrogen bond acceptors, number of heavy atoms, like very easy stuff. But it converts the data point from a string, which has no chemical meaning, other than, I mean, a model can interpret it, but for all intents and purposes, does not have chemical meaning, to a descriptor vector where the elements are explicitly defined chemical compounds. Okay. So then we create this space where the dimensions correspond to, well, actually, we perform PCA rather. And then we project these data points into that reduced space, which is still 120 dimensions. Then we use k-means and we cluster the space. And what this does is it allows us to group molecules that are chemically similar. So molecules in the same cluster are physico-chemically similar. Does this make sense? Okay, cool. So then what we do is we sample 1%. There are a lot of clusters, like over 100, or actually, they're exactly 100. Yeah. So we sample randomly 1% of the data points from each cluster. We dock them and we score them. And then we represent each cluster with the mean score of the 1% that were sampled. Then we want to create a fine-tuning set to further train the model. We sample from the clusters proportionally to the scores of the molecules we score. Okay, so our grand assumption is you only need to sample 1% from the right regions iteratively to align a model closer to that region. So we do this 5 times, and what we find is it works very well. So let me go. Yeah, okay. So there's this protein called CABL kinase. It's implicated in a type of cancer, type of leukemia. And we use it as a case study here because it has known FDA-approved drugs already. So it already has molecules that we know for sure bind to. Okay. So I think there are 7. Yeah, 7. Imatinib, nilotinib, these things on the bottom here. So, okay. Iteration 0. What this is showing here is these white dots are the known drugs. And by the way, this is a 2D representation of chemical space. These white dots are the known drugs. And this here is the molecules generated by the generative model. Okay. And now what we show, and then going to the right is we do 1 iteration of the active learning method, 2 iterations. And what we find is the generated ensemble converges toward the known binders here. So it successfully finds the region of space containing binders without knowing of their existence prior. And now if you look at a comparison, this is the top one is the approved drug, like the actual thing. And then in each panel, the bottom one is the most similar generated molecule to that drug. And what we find is we generate these 2 exactly, and we generate very similar ones to these. Now, these are the most similar ones in the data set, so like don't get it. It's not like it's predicting one, and it's this. But nonetheless, we show that the ensemble evolves toward the region with the known binders. Does anyone have any questions?
This is very nice because it generated distribution that would necessarily outperform the distribution of the training set.
Yeah, no, it does. Yeah, it does significantly. And at a conference I was just at, there were people that were trying to do similar things, and they're sampling like 100,000 compounds to dock. Like it takes way too long. Like, just to be clear, even sampling 1% on an A100 GPU is taking us like 9 hours in iteration. So it's still like not super fast. But like, just imagine like to have to do 1,000 times more than that or 100 times more. It's not happening. It's a difference between like 9 days and like 3 months or I don't know, something crazy. So, yeah, so we also apply it to a protein with no known small molecule binders, the H&H domain of Cas9. Yes. And just pay attention to the C here. So like here is at the beginning, didn't apply it, and this x-axis here is a docking score, you could say, or like it's an approximation to binding affinity. We apply the method and it shifts the distribution to the right. And this has no known drugs. And we're actually collaborating with George Lisi at Brown, who's going to make some stuff. We're going to say, hey, we think this may bind to H&H. Can you make it? Or can you buy it, test it? So, yeah, we're working on this. I think that's probably okay.
Maybe you want to say one word about what is H&H?
Oh, yeah. So, all right, you're the bio person. You guys mentioned. There's this enzyme called, it's a system called CRISPR-Cas9. That's incredibly interesting because it makes possible the ability to basically edit DNA. Now, the way it works is there's this enzyme. There's an enzyme, which is protein, and then there's an RNA sequence, which you engineer. You make the RNA sequence do whatever. Now, say you have some genetic sequence, hypothetically, in you that has a mutation, and the mutation is going to cause all sorts of problems, so you want to remove it. So you make an RNA sequence corresponding to the mutation, corresponding to the sequence with the mutation, and you give the RNA sequence and Cas9, you send it into your body. You can't do it this easily because it's going to cause a few problems, but like in a vial or something, okay, or whatever that they use, the petri dishes or. And what happens is the Cas9 will snip that part of the DNA. This is its function. H&H is a domain of Cas9 that cuts the target DNA strand. So it would be very interesting if we can modulate its function, because one of the main problems with Cas9 is it will make off-target SNPs, which is a big problem. Like you can't.
Third arm.
Yeah, it's gonna cut. You can't just like put it in. It's cutting wrong and stuff. Like this is definitely problematic. But we actually in Victor's lab, we've been collaborating with people at Brown. And what we showed is there are specific mutations that you could introduce into H&H to make it more specific for the target DNA strand. So we're thinking, okay, if we can modulate its behavior with mutations, maybe we can modulate its behavior by finding small molecules. So that's what motivated this now collaboration that's basically starting now, which is to propose potential small molecule binders to H&H to see if we could modulate its function.